there are, in fact, both human and animal evidences for an involvement of neuroinflammation in Parkinson's disease. In humans, epidemiological studies shows that the people who take anti-inflammatory drugs have a lower risk of developing Parkinson's disease. This is not true for all the anti-inflammatory drugs. It is especially true for ibuprofen. And this has been shown in meta-analysis. There is a dose effect. It has been shown also in prospective studies. So it's a relatively strong evidence. In terms of genetic analysis, in terms of risk factor, the GWAS analysis shows that one locus in the HLA-DR big locus is also a risk factor. So there are both environmental and genetic evidences. But these are just susceptibility factors. It doesn't tell you that neuroinflammation is really occurring in the brain and is instrumental. So we need other data, and we have this data. Postmortem studies show that activated microglial cells are present post-mortem in the brain of PD patients, but also lymphocytes, which come from the periphery, from the blood circulation, and infiltrate into the brain parenchyma. And we have shown that CD4 lymphocytes penetrate into the brain parenchyma. But again, this doesn't prove the role of these events. And several groups, including my own group, have switched to animal model to show that either the activated microglial cells or the T cells play a deleterious role and are involved in the degeneration of dopaminergic neurons in the substantia nigra in those animal models. But it is not so simple, because you have also a time course in terms of neuroinflammatory processes. And recently we have published some data showing that at the beginning of the degenerative process, you have some markers which are expressed, some other markers are expressed during the whole neuroinflammatory process, and some are trying to combat the deleterious effect and might in fact be anti-inflammatory, but are involved in this whole cascade. So this means that in terms of neuroprotective strategy, you should not target the neuroinflammatory processes globally, but target the deleterious effect of the neuroinflammatory processes, and at the right time during the evolution of the disease.